Hi, welcome to the screencast. This is a quick review of the neuromuscular junction before we get into the specifics of excitation contraction coupling. Before we talk exactly about the neuromuscular junction, we should review membrane potential. So, what you need to know is that all of your cells, including muscle cells, have what is called a resting membrane potential, which is somewhat negative. This membrane potential is measured in volts. If it's really small, we use smaller units called millivolts. So the resting membrane potential is about negative 100 millivolts, which means there's a small amount of negative charge built up on the inside of the membrane, so it is negative on the inside relative to the outside. There is a separation of charge across the membrane, and in this state, then we say that the membrane is polarized. Just like our globe has a north and a south pole, if you have a positive and a negative end or side to something, then you can say it is polarized. If this voltage is reduced, if there is less of a difference between what's on the inside and what's on the outside, if it becomes, as this says here, less negative on the inside, then we call this depolarization. The last thing that you want to remember about muscle cells and all cells is that thanks to our friend the sodium potassium ATPase, sodium levels are always higher outside of a cell than inside of a cell. And there we go, having trouble with, what do you call that, PowerPoint. So this is just a list of terms. I think they are all in the objectives, but I'm not sure. These are things that you want to be familiar with. I don't necessarily ask specific questions about them on the exam, but you want to be familiar with these terms. Motor neuron, synapse, acetylcholine, which is the neurotransmitter we'll be talking about, motor end plate, which is part of the sarcolemma or muscle cell membrane, receptor, ligand, and acetylcholine receptor, which is a ligand-gated ion channel. Alright, so the neuromuscular junction is where a motor neuron meets a muscle cell, and this is specific for skeletal muscle. So your skeletal muscles are controlled by motor neurons. These are them right here. They arise either in the spinal cord or in parts of your brain stem, and they extend through peripheral nerves either to skeletal muscles or through cranial nerves that come out of your brain and brain stem, and then end at different skeletal muscles. Where a nerve meets its target tissue, whether it's where the nerve meets muscle or where two nerves meet each other, this is called a synapse. So the neuromuscular junction is the synapse between a neuron and a muscle or skeletal muscle. So when you are looking at the neuromuscular junction, at the end of the motor neurons is this bulbous thing that we call a terminal or axon terminal. All you need to know about it for now is that the neurons, or your motor neurons, I should say, contain within them a neurotransmitter called acetylcholine. And when those nerves are stimulated, acetylcholine is released by these vesicles into the synapse or synaptic cleft. So this is the neurotransmitter that is going to stimulate your muscle cells. Then, if we look at the muscle cell, this section of the membrane is what is called the motor end plate. The motor end plate is another tricky technical term for cell membrane. It's the part of the cell membrane that's right underneath the neuron. So it's part of the sarcolemma, which is another one of these stupid anatomy and physiology terms we have for the cell membrane of a muscle cell. So the portion of the muscle cell membrane that you find underneath a neuron is called the motor end plate. And remember, like all cells, it has a somewhat negative resting membrane potential. So you want to envision some negative charges lined up here on the inside of the membrane. When acetylcholine is released into the synaptic cleft, it is going to stick to what is called acetylcholine receptors, These are ligand-gated ion channels. So a receptor is anything that detects stuff, as it says right here. And then a ligand is anything that is detected or anything that sticks to a receptor. So these two terms kind of define each other. Receptors stick to ligands and ligands stick to receptors. So in the case of acetylcholine, this is a protein that I should say, in the case of the acetylcholine receptors, they are proteins that also have ion channels within them, such that when acetylcholine sticks to the extracellular surface of this protein, it pops open and a little channel forms that sodium is able to flow through. If you remember from the first slide, sodium levels are always high outside of cells relative to inside of cells, so that when a sodium channel opens, sodium is always going to flow into the cell. If sodium flows into a cell, then that is positive charge that's entering the cell near the cell membrane. This is going to make the inside of the cell membrane less negative or more positive, so it's going to depolarize the cell. So this just sets us up for understanding excitation-contraction coupling. Thank you for listening.